My name is Susan McEvoy, and I'm a PhD candidate at the University of Connecticut. Today, I'll be talking about beech bark disease resistance and variation and assembly error in two Fagus grandifolia genomes. Fagus grandifolia, or American beech, is the only Fagus species native to North America. Found in the eastern US and Canada, it's very common in certain forest types, such as beech, birch, maple. Beech nuts are edible and provide food for many animals. The bark is very thin, but the wood is hard and used for a number of products, such as furniture. Beech bark disease, or BBD, was introduced in Nova Scotia from Europe around 1890. In forests that have been affected by BBD for a long time, about 1% of the trees remain disease-free. It is a scale insect fungal complex. Top left here, we have a tree with signs of the disease. Bottom left are the fruiting bodies of the fungus, and right is the cankered stem. There's now a newer threat called beech leaf disease. Nematodes are the primary cause there, and it was first seen in Ohio in 2012 and is rapidly spreading. Here we have the native range of American beech per the BN database. This is showing the spread of BBD as of 2019 using Forest Service data. Light gray is state reports and dark gray are county reports. The red area is reports of beech leaf disease as of 2021. For BBD, resistance appears to have a genetic component. In a previous study, transcriptomic markers were used to develop a small genotyping assay that assessed a pedigreed population to develop a linkage map. In addition, a larger number of unrelated individuals known to be susceptible or resistant were genotyped, and a GWAS identified 4 markers associated with resistance in that population. All 4 were located in the same locus with a single gene, which was annotated as metallothionein in European beech, the only Fagus genome. There is an established breeding program that has had success with breeding resistance, but both this and breeding for beech leaf disease resistance would benefit from more genetic resources. Development of the American beech genome was originally initiated by 2 separate research groups, our lab at UConn and a group at Boyce-Thompson Institute at Cornell. On discovering this, we joined forces to compare the 2 reference genomes. The Cornell beech is known to be resistant, and the status of ours is unknown as it's in a region that has not been exposed. We each started with very different sequencing coverage. UConn's was very high, approaching 200x even after filtering, while Cornell's was much lower. Their approach was based on mini-ASM followed by error correction. Ours was Mazurka, which contains an error correction step. Both were filtered for haplotypes and scaffolded with all maps using the genetic map I mentioned earlier. To do a more complete comparison of assembly methods, we swapped sequencing and reran everything for a total of 4 assemblies. In spite of the very different sequencing coverage, assembly characteristics had trends by workflow. The first 2 here are Mazurka. They didn't change much in size between draft assembly and scaffolding. Busso scores improved slightly at around 96% total complete. Number of contigs dropped by about half, and N50 improved greatly to around 25 megabase. Mini-ASM was different in that it appeared to assemble both haplotypes, which were then filtered out. We can see this in the length and the Busco duplication nearing 50%. Final Busco scores are low, especially for Unicon mini-ASM at 70% complete. The resulting number of contigs was low, but there's fragmentation in terms of the high number of gaps represented as N's. Missing and fragmented content is also reflected in the gene totals, where the Mazurka assemblies are similar, but the total decreases to 20K in Yukon mini-ASM. The 2 Mazurka assemblies were compared to Fagus sylvatica, which is a chromosome-level assembly. Blocks of synthetic genes had good macrosynteny in sylvatica compared to both Mazurka assemblies and those with each other. So the question is, with 4 genomes from 2 individuals, can we more accurately detect assembly error, true biological variants, and beech bark disease alleles? Transitioning into SVs and assembly error, there are a tremendous number of SV calling tools available. There are different approaches involving alignment of long reads, short reads, and an assembly to assembly. Tools have different strengths and weaknesses that can be better at detecting different types of SVs or different lengths. So we picked a mix of popular tools from the different approaches that seem to score better in benchmarking studies. We ran these using alignments of Yukon reads to both Cornell assemblies and Cornell reads to Yukon assemblies. The results were filtered depending on the tool. So Survivor combined these sets and merged any call by more than one tool. We reran the read alignment tools with assembly reads back to the same genome, so a self-to-self alignment. Assembly reads should map back to the genome well, so any variants called indicate assembly error. We combined those sets and then used Survivor to compare the merged assembly error set with the merged SV set and filtered out any error found in the SVs. Here are SV counts for the assembly error and the different approaches, long read, short read, and assembly. While there is substantial overlap, you can see there are still a large number unique to each approach. And here's another view of the results after assembly error was filtered out. And here are the results for each tool within the different approaches, assembly to assembly, which had strong overlap, long read and short read. Lumpy had quite a few unique calls, even after some filtering, excluding regions of high coverage. And here we can see a breakdown by SV type, deletions, duplications, insertions, inversions, and translocations. You can see that the Cornell assemblies resulted in the most SVs. For assembly error variants, Yukon Mazurka had the least error, and Cornell Mini ASM had the most. Yukon Mini ASM had such a low BOOSCO and total length. I would have expected that one to have the most error, but since this is just counts and doesn't factor in size of SVs, they could contain fewer but larger SVs. We used the assembly error to validate scaffolds that were split by all maps during scaffolding and placed on separate chromosomes. Here's one example of a scaffold that was split and put on chromosome 12 and 2. These are snapshots of the ends of the scaffold fragments to examine read alignment and assembly error SVs. Alignment on the outer edges was poor. Alignment on the inner ends is good. And the read pairs and the supplemental alignments match between the two. There are also translocation SVs at this point. So AllMaps has introduced misassemblies here, but the larger placement on chromosomes seems okay, as the genes within these regions match synteny with the overall chromosome. So for example, chromosome 12 matches with the European Beech 1, and chromosome 2 matches with 3. We also checked the error around the Beech bark disease locus. This is zoomed out from the locus quite a bit, and you can see the markers in the center here, in the top track. It's the large assembly error SVs on either side. Those are ARCSR deletions. The bottom track is SVs called on long read sequencing from a susceptible individual. The assembly error has been called here as SVs, but because we have identified it, we can filter these out from the final results. Now let's zoom in on the actual BBD locus to look at the variants within. The region was identified in chromosome 5 of the Cornell Mazurka assembly by the markers that were identified in the transcriptomic association study I mentioned earlier. So first, let's get oriented to the tracks you'll see throughout the next few slides. The top are assembly long reads aligned back to the Cornell reference, so self-to-self alignment. And we have assembly sequence, the resistance markers here, gene annotation, and repeats. So we can already see clues of both SNPs and SVs that differentiate resistant and susceptible alleles and the way they are split rather equally amongst the reads as visualized in the coverage track. First, we'll look at a few example SNPs to get a better idea. At the top, we've added long read alignments from the susceptible individual. The next set are those assembly read alignments, and here are our markers that contain the SNPs of interest. So the central SNP for the marker ending in 126 here, it has all 38 reads SC in the susceptible alignments. And in the assembly read alignments, 54% match susceptible and 46% are G. The SNP for the marker ending in 406 is nearly all susceptible, so this might not be the best marker for identifying resistance. Just to the left of it, we have a better option with 48% Cs matching susceptible and 52% resistant. The SNP for a marker ending in 334 is okay, but it's not clear-cut in the susceptible individual with a 65 to 35% split. And now the SVs, there are 2 deletions in the susceptible individual. In the assembly reads, similar to the SNPs, about half the reads have the same deletion, seen here where the long read coverage drops by half. SV calling of the susceptible individual found 3 SVs in this area, but there isn't actually much support for the third among the read alignments. Here we have the same region in the Yukon-Mazurka genome, also chromosome 5 and the same tracks shown here, susceptible alignments, assembly alignments, markers, annotation, and repeats. The lighter colored read alignments indicate lower quality mapping for both the assembly reads and susceptible individual. The markers also mapped equally well to an unplaced scaffold, number 236. The read alignments were better quality, where chromosome 5 assembly sequence in both the Yukon and Cornell variants indicated resistant allele. This assembly sequence seems to be susceptible in terms of the SNPs of interest. But we still see heterozygosity in the assembly read alignment. Now going back to the mini ASM assembly, which seems to assemble both haplotypes of everything, the locus markers are mapped back to both a resistant and susceptible allele. So it seems that Mazurka collapsed the haplotypes into a resistant consensus in the Cornell assembly, but both remained in the Yukon, here in the form of scaffold 236. Perhaps the difference in sequencing coverage has something to do with it, given that the locus is otherwise very similar in both genomes. So to wrap up, we've identified variants that seem to be associated with resistant and susceptible alleles. Both the Cornell and Yukon individuals appear to be heterozygous, and since the Cornell individual is resistant, then perhaps the Yukon one is as well. The R0 S20 susceptible individual matches the susceptible allele. There are not short read resequenced resistant individuals I didn't cover here, which match the resistant allele. Now we have 2 genomes and broader sets of SVs for future beech bark disease or beech leaf disease, resistance breeding, or other analysis. I'd like to thank everyone involved at BTI, especially Adrienne Powell, who worked with me a lot on selecting and running SV calling tools and BBD locus examination. Jose Cirque and Nate Swenson for providing the sequencing UC Yukon, and thank you.